Welcome to the Health Headliner podcast by Todo Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine trying to understand the wiring of a city with a map that's, constantly getting things wrong. Streets that suddenly end, maybe buildings misplaced. That's the incredibly complex challenge neuroscientists face when they try to map the vast network of connections within the brain.
It's a huge undertaking.
But what if there was a way to automatically find and fix those errors, to give us a much clearer picture of our mind's true organization?
That's exactly what we're digging into today. We're taking a deep dive into a new piece of software called neural decomposition.
Neural decomposition, okay.
Yeah, researchers at Baylor College of Medicine developed this tool to tackle the very time-consuming job of correcting errors in the enormous data sets that map how our brain cells connect.
Okay, let's unpack this a bit. We hear about these amazing efforts to map the brain, right?
Right.
But the idea that these maps might have significant errors isn't something that always comes to mind. Why is it so vital to get these connections right in the first place?
Well, it's absolutely fundamental. Fundamental to understanding how the brain achieves its remarkable abilities. Things like problem solving, learning.
The really complex stuff.
Exactly. The specific ways our brain cells, our neurons, are wired together, the patterns of these connections, that's what enables these functions. Even the most advanced AI, artificial intelligence systems haven't yet fully matched the brain's efficiency.
Right.
So having an accurate blueprint, a correct wiring diagram, is just essential to understanding the biology underneath it all.
Okay. So for this deep dive, you've looked at a health news article from April 9th, 2025, talking about this neural decomposition software.
Yep, and the detailed scientific paper. Published in Nature, same date.
Got it. So what were the main goals of the researchers? What were they trying to achieve with this new tool?
Well, the primary goal, and this comes through in both sources, was really to create a reliable way to automatically detect and correct mistakes.
In these massive brain map data sets.
Exactly. Huge amounts of data coming from these large-scale projects. And secondly, they wanted a tool that could then use this cleaner, more accurate data to reveal new insights into how the brain is structured, how it functions. But, to really grasp the challenge here, it helps to understand the kind of data they're actually working with.
Absolutely. You mentioned large-scale projects. One name that keeps popping up is machine intelligence from cortical networks, or micron.
Oh, yes. Machine intelligence from cortical networks. That was a huge 7-year collaboration.
7 years. Wow.
Yeah. Over 150 scientists involved. They worked on creating the most detailed map we have of a mammalian brain.
Which part specifically?
A tiny cube, just 1 cubic millimeter of the mouse visual cortex.
A 1 cubic millimeter. That sounds tiny, but I'm guessing the detail is immense.
Oh, the scale is staggering. This single cubic millimeter contained over 500 million synapses.
500 million connections.
Connections, yeah. Miles of axons, those are the long fibers transmitting signals, and over 200,000 individual cells, all packed in there.
That's just hard to even picture.
It really is. And what made the machine intelligence from cortical networks project especially valuable was that it didn't just map the physical connections.
Right.
It also gathered functional data. They actually recorded the electrical activity of these same neurons while the mice were shown different visual things.
Oh, so linking structure to function directly.
Precisely. It allowed researchers to link the physical structure, the wiring diagram, to how these neurons actually communicate and process information.
So they're getting both the blueprints and how the system actually works. How do they even generate these enormous data sets? It sounds incredibly technically demanding.
It requires a monumental effort, seriously. Researchers take incredibly thin slices of brain tissue.
Like super thin.
Extremely thin. And they image them using a powerful electron microscope.
Okay.
These two-dimensional images, millions of them, then have to be meticulously aligned and segmented.
Segmented.
Meaning they trace out each individual neuron and its connections through all those image slices to reconstruct the full three-dimensional structure.
Okay, I'm picturing like trying to piece together an unbelievably complex 3D jigsaw puzzle from millions of tiny photos.
That's a really good analogy, and it takes an immense amount of time, even before you start worrying about mistakes.
Which brings us back to the errors.
Exactly. These initial reconstructions, while incredibly impressive feats of engineering and computation, often contain errors.
Like what kinds of errors?
For instance, Parts of two completely separate neurons might be mistakenly joined together. Those are called merge errors.
Merge errors, got it.
Or the opposite can happen. A single neuron could be broken into multiple separate segments. Those are known as split errors.
Okay, merges and splits. And these aren't trivial mistakes, I assume.
Not at all. If these errors aren't corrected, they can lead to a fundamentally incorrect understanding of the brain's wiring. Completely wrong conclusions. And that's where neural decomposition comes in.
Right, the automated proofreader. So how does it actually do that? What's the underlying process?
Okay, so neural decomposition takes the complex three-dimensional shapes of neurons, these intricate meshes, and it simplifies them. It turns them into more manageable, annotated diagrams or graphs.
So like turning that tangled web we talked about into a clear, organized chart.
Exactly. A chart with labels for all the key parts, the main body of the cell called the soma, the branching fibers, axons and dendrites, and even the tiny bumps on dendrites called spines.
So it's like taking a really intricate circuit board and creating a simplified schematic. That makes it easier to analyze.
Precisely. By creating these diagrammatic representations, the software can then analyze key characteristics of the neurons.
What kind of characteristics?
Things like the size and shape of the soma, how the axons and dendrites branch out, the presence and form of those spines. By analyzing these features, neural decomposition can automatically identify common errors, especially those merge errors where two neurons have been incorrectly joined.
That's interesting. Can you give a more specific example? How does it actually spot a merge error? What's the clue?
Sure. One important indicator is a sudden, sort of unnatural change in the thickness of a neural fiber where two segments connect.
Ah, like if a pipe suddenly got way wider or narrower for no reason.
Exactly that kind of thing. It suggests maybe two different pieces that don't belong together got stuck together in the reconstruction.
Okay.
Another thing it looks for is when an axon branch suddenly turns sharply back towards the cell body. That's generally unusual biologically.
So it's using these rules of thumb based on what we know about neuron structure.
Yeah, they call them heuristic rules. Basically, logical checks based on neuroscience knowledge about how neurons are typically structured, used to flag potential problems in the reconstruction.
That makes sense. It's using biology to check the digital map.
Yeah.
Once it finds these potential errors, does it automatically fix them? Or just flag them?
In many cases, it proposes a correction. The researchers tested how well neural decomposition worked by comparing its automatic corrections to those made by human experts.
The gold standard, but slow.
Very slow, very painstaking. And they found a high degree of agreement, especially in correcting merges between axons and between axons and dendrites.
So it's pretty accurate.
Yes. And using the software's suggestions significantly sped up correcting certain types of merges, like those involving multiple cell bodies, compared to doing it all manually. It highlights likely errors, and then a human can often quickly confirm and make the fix.
So it's not just finding errors, it's really streamlining the correction process, making it much more efficient.
Exactly right. Efficiency is key here. And beyond just proofreading, neural decomposition also extracts a wealth of information automatically.
What kind of information?
About the morphology, that's the shape and structure of neurons and how they connect. This includes details at different scales, from small segments of a neuron up to the whole neuron, and even relationships between nearby segments.
Okay, so we have this powerful tool. It cleans up these massive brain wiring diagrams, makes them more accurate, pulls out tons of data. What are the big implications here for actually understanding the brain? What new research does this unlock?
The implications are really significant for neuroscience research. I mean, making these incredibly large, complex data sets more accurate and easier to analyze, it just opens the door to new discoveries.
How so?
Well, it lets us probe the fundamental rules governing how the brain is wired and how that wiring leads to function. By automating error correction, neural decomposition removes a major bottleneck.
Right, the tedious proofreading step.
Yeah. So researchers can focus more on the why behind the wiring, not just struggling with the what.
What kind of rules are we talking about? Specific patterns?
Exactly. With cleaner data, researchers can more reliably study neural circuits, how different types of neurons connect with each other, the specific patterns or motifs of these connections. Understanding these patterns is crucial for understanding how information flows and gets processed. We can start to see if there are common connection patterns that might be the basis for specific brain functions, like standard circuit designs almost.
So it's like identifying the fundamental building blocks of the brain's communication network, the recurring designs.
Precisely. And there's more. Neural decomposition can also help classify different types of brain cells based just on their shape and connection patterns.
Even without genetic information.
Yes, even without direct genetic markers sometimes. This is important because it allows us to link the structural organization we see in these maps to the different jobs, the different functional roles that various types of neurons perform.
Could you give an example?
Well, we might find that neurons with a specific branching structure consistently connect to neurons with another particular shape that strongly suggests they work together on a specific task or pathway.
Okay. And you mentioned dendritic spines earlier, these tiny bumps. How does neural decomposition help us understand those better?
Right, dendritic spines, they're these tiny protrusions on the dendrites, the receiving branches of neurons. And they are absolutely critical for synaptic plasticity.
Plasticity meaning?
The ability of connections between neurons, between the synapses, to strengthen or weaken over time. It's the fundamental basis of learning and memory.
Okay, crucial structures then.
Hugely important. Neural decomposition can perform detailed analysis of the shape of these spines. It can segment a spine into its head and neck, measure features like size and shape.
So really fine-grained detail.
Yes. This allows for much more detailed studies of how spine shape varies, maybe across different neuron types or in different brain areas, or maybe even under different conditions. It gives us insights into how synapses might be changing during learning, for example.
This is all fascinating, and it sounds like it could have major implications for understanding brain disorders, potentially.
Absolutely. That's a huge potential benefit by enabling a much more detailed understanding of the brain's microscopic circuitry.
Down to the individual connections and spines.
Exactly. Scientists hope to identify specific problems in specific neural circuits that might underlie neurological disorders. Things like Alzheimer's disease, Parkinson's disease, autism spectrum disorders.
So finding the wiring faults, essentially.
That's the hope. If we can pinpoint the precise wiring issues associated with these conditions, it could pave the way for developing more targeted and effective treatments down the line.
And the research also compared data from the mouse brain to some human brain tissue, you said, using neural decomposition.
Yes, that's right. They use the tool to analyze data from the mouse visual cortex, that machine intelligence from cortical networks project dataset, and also from a dataset of human temporal lobe tissue, the HR1 dataset.
What did they learn from that comparison?
Well, it allowed them to highlight both similarities and differences in how neurons are organized across these two species, which is really interesting from an evolutionary perspective.
Any specific examples?
Yeah, they observed differences in where synapses tend to form on a specific part of the neuron called the axon initial segment, and also on the main cell body, the soma. The patterns were different between the mouse and human samples they looked at.
So comparative analysis gives clues about brain evolution, too.
It can, yeah. These kinds of comparisons offer valuable insights.
Okay, so neural decomposition sounds incredibly useful, really powerful.
Yeah.
But are there limitations? Things it can't do right now?
Oh, definitely. It's important to have a realistic perspective. While it's a significant step forward, it does have limitations.
Such as?
Well, the current version is primarily focused on correcting those merge errors we discussed where neurons are incorrectly joined.
Right.
It doesn't yet fully address other types of mistakes, like those false splits, where a single neuron is incorrectly broken into multiple pieces.
Okay, so it's better at fixing things stuck together than things broken apart.
At the moment, generally, yes. Or situations where the initial reconstruction of a neuron is simply incomplete, parts are missing. It doesn't magically fill in missing data.
So still room for improvement and development on the types of errors it handles.
Exactly. Also, remember, the automated proofreading relies on those heuristic rules, the rules of thumb.
Based on typical neuron structure.
Right. And while the testing shows it's quite accurate, there's still a possibility of the software making an incorrect change sometimes. It's not infallible. Human oversight is still valuable.
That makes sense. And what about classifying the different types of brain cells? How reliable is that function?
Well, the accuracy of classifying cell types using the features neural decomposition extracts really depends on the quality and completeness of the initial data.
Garbage in, garbage out, basically.
To some extent, yes. And it also depends on the data used to train the classification models. If the initial reconstruction is noisy or incomplete, or if the training examples aren't great, the classification accuracy might be lower.
Okay. And I imagine the findings we have so far, based mainly on mouse visual cortex and a bit of human temporal lobe, they might not apply equally everywhere.
That's a very important point. The current findings are primarily based on those specific areas. We definitely need more research to see how well neural decomposition works in other brain regions.
Which might have different cell types, different wiring patterns.
Exactly, and also how well it works in other species beyond mouse and human. So generalizability is still an open question that needs more work.
Good to keep those limitations in mind for a balanced view. Now, the sources also included some thoughts from the scientists who actually developed this. What were their main takeaways or hopes for neural decomposition?
Yeah, Dr. Jacob Breimer, who is a senior author on the Nature study, he emphasized that the main goal is really to uncover the importance of wiring in the brain.
Understanding why the connections matter so much.
Right. Understanding the biological mechanisms behind the brain's extraordinary problem-solving capabilities. He believes neural decomposition will be essential for advancing our understanding of neural networks as we get more and more of these detailed brain maps.
He sees it as a key tool for future discovery.
Absolutely. And Dr. Brendan Slee, the lead author,
What was his perspective?
he highlighted how neural decomposition tackles this really difficult problem of automatic neuron proofreading by breaking it down into simpler steps.
How so?
Steps that are kind of similar to how a human expert would visually inspect a neuron. He pointed out that even identifying features like those dendritic spines we talked about can help infer the cell type, which in turn makes the proofreading process itself more manageable and potentially more accurate.
So it's about making a hugely complex task more achievable, more scalable for researchers everywhere.
Exactly. And the researchers, they explicitly stated their hope that neural decomposition will help more researchers work with and extract insights from these massive Connectomics data sets.
Democratizing the data, in a way.
Yeah, making it more accessible. They really see this as marking a new era in neuroscience, where machine learning tools and biological understanding are increasingly integrated, working together.
It certainly sounds like a powerful catalyst, with a lot of potential to push our understanding of the brain forward.
For me, yeah, neural decomposition represents a really significant step. Not just mapping the incredibly detailed wiring, but making sure those maps are accurate and then actually extracting meaningful information.
Tackling that bottleneck.
Addressing that major bottleneck, the slow, tedious error correction. It just opens up exciting new possibilities for understanding brain organization and function. It makes these vast data sets more accessible, letting us ask and hopefully answer increasingly complex questions.
This has been a really fascinating deep dive into neural decomposition and its implications. It highlights how crucial these sort of behind-the-scenes tools are for making progress in neuroscience.
Absolutely. It's a great reminder that major scientific advancements often depend just as much on innovative tools and methods for handling and analyzing the data, especially when it's this complex.
So as we've seen, neural decomposition represents a significant advance, a big step in our ability to map and understand the intricate wiring of the brain. It's a tool that, not only speeds up a crucial but pretty tedious process, but also lets us ask and answer more complex questions about how our brains are put together.
Yeah.
We definitely encourage you to explore the original research paper in Nature and the news article from Baylor College of Medicine if you want a deeper look into the specifics of neural decomposition and that machine intelligence from Cortical Networks project. So, thinking about all this, what aspects of brain connectivity are you most curious about now that tools like neural decomposition are making these intricate details more accessible? Something to ponder.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.